Hi, welcome back to the Medicinal Plants Trail. This time we're looking at how Taxol works. Cell division, control versus cancer. Let's get started. What is cell division? Cell division is a process by which organisms grow new and replace old cells. Cell division occurs in two distinct phases. Interphase, a non-dividing state where the cell grows and replicates its DNA, and then mitosis, the dividing state, where the nucleus and then the cell itself split into two. Let's look at these in a bit more detail. Firstly, interphase. The cell grows and then duplicates its DNA, but the chromosomes are still in their decondensed state. Then we enter mitosis, which is split into a further 4 phases. Firstly, prophase. Here, the chromosomes condense into pairs of sister chromatids. Then, special spindle fibers for maneuvering the chromosomes form. Then the nuclear membrane begins to break down. Next is metaphase. At this point, the nuclear membrane is completely dissolved. The chromosomes have been captured by the spindle fibers and aligned at the cell's equator. Here we have a very important stage called a spindle assembly checkpoint. At this point, the cell confirms that all of the chromosomes have been properly attached to their spindle fibers before proceeding with mitosis. Next is anaphase. The sister chromatids are separated by the shortening spindle fibers. The spindle fibers also pull against each other, which lengthens the cell. Finally, telophase. The chromosomes then arrive at the poles, and the nuclear membranes reform. In their new nuclei, the chromosomes unwind back to their decondensed state, and then cytokinesis divides the cell into 2 identical daughters. What happens in cancer, though? Occasionally, mistakes happen during cell division. Normally, the cell's checkpoints are able to pause the replication process until the mistake has been fixed. However, sometimes mistakes are not always spotted at checkpoints. And if mitosis continues and a mutated cell is produced, the mutated cell can go on to divide rapidly, forming a tumour which can turn cancerous. So how does Taxol work to help treat cancer? Taxol works by solidifying the structure of the spindle fibers in the rapidly dividing cancer cells. Unable to move, the spindle fibers cannot divide the chromosomes into the 2 sister chromatids, allowing them to be divided between the 2 daughter cells. This traps the cell at the spindle assembly checkpoint and signals the cell to undergo programmed cell death, known as apoptosis, which prevents any further harm from occurring from the damaged cell.